So Thaís is an assistant professor at the University of São Paulo, and her primary research interests are soybean anthracnose and Myrtle Rust. And today she'll be talking with us about using mating compatibility gene markers to detect new incursions of Austropuccinia psidii. So Thaís, if you're ready to get started, feel free to get underway.
Okay. Hi, hello. Thank you, Madge, for having me here. So I'm Thaís, and I'm here today to talk about some genome sequencing of ACDI strains and how we are using these new sequences in different lines of research within our group, including the use of mating compatibility gene markers to detect new incursions of Austropuccinia psidii. This is a joint project within our university in Brazil and the Australian National University, the University of Sydney, and several other institutes or universities in Australia. It started with me going to spend time in Benjamin Schressinger's lab last year. So I would like to thank all our collaborators along this project, and also the financial support of the Australian Research Council and FAPESC. Before we start to talk about genome sequencing, I just want to remind you about the world scenario of ACDI distribution. So over the last few years, some works mostly based on microsatellite markers or some biological works related to the presence of two polyphagous biotypes, meaning they are able to infect hundreds of different hosts within the Myrtaceae family. One restricted to South Africa, it's called the South African biotype or genetic group. And the other one, it's widely spread around the world and is responsible for this exponential spread and incursions of Myrtle Rust. And it's the only one present in Australia and New Zealand, for example. While this pandemic biotype is widely spread, in the center of origin of the disease, that's Brazil and South America, it was only detected in Colombia so far. And we have a more diverse population situation there. So we have the presence of multiple genetic groups associated with different host species. We have commonly present one biotype associated with guava, one with rose apple, one with Eucalyptus, and sometimes these two groups are the same. So our first question here is to try to understand what are the genetic traits associated with the rapid global dissemination and establishment of the pandemic biotype of ACDI. Because first we need to comprehend in order to manage the disease. So to address this question, we started this work by selecting these isolates for sequencing. So we started with two single postal isolates, one from rose apple and one from guava. We inoculated these isolates in the original host species, characterized the monocycle of the disease, and find out that at the end of the experiment, they were able to complete the asexual life cycle by sporulating in these hosts. At the same time, these isolates were cross-inoculated in the other two, in the same two host species. And after the end of the experiment, we found out that only some necrotic lesions were present in the host surface, and sporulation did not occur. So the life cycle the asexual life cycle was not completed. And we confirmed that by analyzing these leaves on confocal microscopy. You can check more details on this work in this publication from last year. So we selected this rose apple and guava isolate, along with one Eucalyptus isolate that's very well characterized over the years here in Brazil. and to do the genome sequencing. And to do this genome sequencing, we use the heterogeneous 4 stage of the life cycle that's characterized to be dikaryotic, meaning it has 2 haploid nucleus inside of each heterogeneous 4. And this stage is the one responsible to cause these common symptoms we can observe in the field. We went through some DNA extraction, cross-linking RNA extraction, and sequenced these isolates in different platforms with long reads and high C reads. And we used these generated data across extensive bioinformatic pipeline. And we ended up with 3 phased chromosome level assembles for the 3 resilient isolates. And we also reassembled the pandemic biotype genome. That's the last one here. So each big circle here represents 1 isolate. And inside of these circles, we can see 2 smaller circles that represent the 2 haploid nuclei that we are calling here haplotype A and B. And inside we have a number that means the number of chromosomes that we were able to assemble for each of these nuclei. Together, these 2 nuclei inside each isolate have around 2 giga size and are composed by 88% of transposable elements. We also assembled the mitochondrial genomes for guava and rose apple strains, while the other 2 were already available in public databases. And together, these represent the 4 largest fungal genomes that were assembled thus far. So now I will talk about some advances that we are having using these genomes and some future directions. So this is a work in progress. We still have like a year to finish all the analysis. As you may have noticed, we have 2 strains here, the Eucalyptus and the pandemic strain, that have different numbers of chromosomes inside of each nuclei. So we want to use these new resources to allow to answer some fundamental questions in fungal biology. And we were able to see this unique characteristic when we check the HiC contact maps for these genomes. So we phased these genomes at the same time. And here we have the HiC contact maps of the dikaryotic assembly. So each big orange square represents a nucleus. So here we have haplotype A and B, and each blue square inside this nucleus represents 1 chromosome. So for haplotype A, we have 19 chromosomes, and for haplotype B, we have 17 chromosomes. And the diagonal lines here represent the linkage between the chromosome for 1 nucleus to the homologous chromosome in the other nucleus. And we can see here that chromosome 14 doesn't have any linkage with haplotype B, and therefore we have 2 copies linked inside haplotype A. Now we already checked, this is the pandemic biotype genome. We already checked if these chromosomes are the same that are swapped in the Eucalyptus genome, and they are not. And now we are trying to figure out if these maintenance of 2 chromosomes inside the same haplotype have any evolutive advantage. So we are going through this analysis right now, but I don't have an answer yet. Next, these new resources may allow us to find to answer our very first question, that are the genetic traits associated with rapid global dissemination and establishment of the pandemic biotype of psidii. And we are still starting with this analysis. We did some comparative genomics, but the only thing that's ready so far is the factors, candidate factors characterization. So we define the factors here as secreted proteins that facilitate infection by suppressing plant defense responses and alter host cell structure and function. So we characterized, here we have the 4 strains, and we characterize the candidate factors separately for each haplotype within each strain. So each line here in the graphic represents one haplotype. And compare those candidate factors with other 9 Rust species. So as you can see here in light blue, most of the candidate factors are present in all the 4 strains of psidii, but are not present in other Rust species. So these are probably psidii specific based on our data set. So those are potentially related to the specificity to the Myrtaceae family. Next, we have some candidate factors that are specific to each psidii strain, meaning they are not present in other strains of the same species, neither on other Rust species. And these are potentially related to the preference to different host species. Now we will check on our transcriptomic database if those candidate factors have some evidence of expression in planta, but also we'll do some more extensive analysis in transposable elements and their role in the evolution and expansion of these pathogens. Lastly, we are also using these resources to develop new tools that may potentially be used for biosecurity applications. So to do so, we are using sexual mating type genes to differentiate the pandemic biotype from other strains of psidii, as these other strains represent one of the most serious threats to Australia natural environment, for example. So in psidii, mate compatibility is governed by a biallelic PR gene, represented here, and multiallelic HD genes. And we want to use these multiallelic HD genes to differentiate these exotic strains from the pandemic biotype of psidii. The first step of this work was to check if this HD locus is multiallelic and check if there is variation within different strains. So at the time, we had the pandemic biotype genome, the Eucalyptus data for these alleles, and the South African genome data for these alleles, all of them publicly available. We check that they are variable across haplotypes and strains. So here we have the representation of the 2 alleles that can vary across them. And we confirm that by extracting the HD alleles from the rose apple and from the guava genomes that we assembled. We did a phylogenetic tree for BW and for BA. And you can see here that for each isolate, we have 4 alleles, 2 of each. So the pandemic isolate, for example, has allele BW1 and BW2. And here for BE, we have BA1 and BA2. And to be considered the same strain, all the 4 alleles need to be more than 95% identical. So the pandemic strain is represented by this variation of dark and light green. We then designed some degenerated primer pairs. Here in blue, one primary pair for the BW allele, and in pink, one for the BA. And if we use the most forward and most reverse primer, we are able to amplify the whole loci locus. So this this primary design was based on this consensus sequence of the alignment of these different strains. Then the next step was to check if we could apply this HD loci for detection of new incursions. So therefore, we use positive controls that are the same strains that had their genome sequence. So for rose apple, Eucalyptus, the pandemic one. And we can see here, based on ITS and other mitochondrial genes, that there is no variation on phylogeny for these three strains. All of them are considered the same species. We also used some single pustule isolates from rose apple too, and for Syzygium samarandense that were collected in São Paulo, Brazil, and some field samples also from São Paulo from 3 different hosts that were extracted from oridinospores collected from these leaves or directly from infected leaves, and also a field sample that was collected in Australia, in Sydney. These Brazilian samples were characterized, strains were characterized before in another publication, and we can also see that based on ITS and the ITS, we don't have any variation for the species. So after amplifying, we extracted the DNA, amplify all of them with the primer pairs, and sequenced them on Oxford nanopore technology. So the diagnostic test works with the amplification, followed by the sequencing of the amplicon. The sequenced amplicons were reconstructed, and for each strain, we ended up with 2 BW and 2 BA alleles. And in the example here, we have on top, the positive control for the pandemic biotype, followed by the Sydney sample, that's a field sample. And we can see that for the 4 alleles, BW1, 2, BA1 and 2, we don't have any variation. So the reconstructed alleles with more than 95% of identity were considered the same. And this was confirmed by lineage calling based on phylogenetic trees again. So we can see here that all the 4 alleles collected from field samples from Sydney clustered with the pandemic biotype. And all the samples that came from Brazil belong to different lineages or strains. So to be continued, as I mentioned, this is a work in progress. We are still working in all those lines of research. Thank you for listening. And if you want to know more about our group in Brazil, you can scan this QR code. We are working with some epidemiological components and control of tropical and temperate rusts on the global climate change scenario. Thank you.
Thanks, Thaís. That was a lot of work and just really, really incredible. So I'm happy to field questions from the audience. So audience, if you have any questions, just put them into the chat and then I will ask them. Okay, so the first question that I will ask, because it's just so interesting that you're finding these differences between the different strains, and just I was wondering if you could just talk about like the the advantage of whether we should keep them as Austropuccinia psidii, or whether do you think that, you know, having like a more narrow species concept would be better?
I think the tricky thing about this is to define a species, really, because based on the common markers that are being used and based on the whole mitochondrial genome, everything's still the same species in the phylogenetic tree. And ACDI is different from other systems like Myrtle Rust, so we cannot actually test all the different more than 400 host species to see if they are form specialis or something like that. So I don't know. Maybe if we compare the full genome, we can see some difference, but how many markers should we use to differentiate the species? I mean, that's tricky. Based on what we have and what we can use as a reference, I don't think it's possible yet. Maybe in the future.
Yeah, no, it is really interesting, and definitely, you know, something that we struggle with, whether you lump species together or whether you split them apart. And
Yeah.
so there's a question about whether the genome assemblies that you've made, are they available as yet?
Not yet. The previous assemble of the pandemic biotypes available on NCI. We should release the updated one in the next few months as we are finishing to write the paper. But the other three, we still need to do a bunch of analyses before we release them.
Yeah. So another question is that it's interesting that there are different numbers of chromosomes in the nuclei of the Eucalyptus and pandemic isolates. Has this been seen before in any other fungi?
Yes, I'm not sure yet, but one of the Myrtle Rusts mentioned it in a publication. I think Puccinia triticina, but I'm not completely sure. And I checked the data, and it has the same phenomenon for rusts. That's the only one I checked, but more assembles, more genomes are becoming available, and yes, we want to check more. But it's also starting to be a description not for 2 copies of the same chromosome in a nucleus, but different numbers of chromosomes in dikaryotic basidiomycetes. But then they are talking about new form chromosomes and something like that in other species of fungus. So we are still trying to figure out why and how does it work. We don't have an answer, like a complete answer yet.
Yeah, no, it's just really bizarre to just think about it, and like, you know, but it's obviously, it's not hampering the success of this particular pathogen. It seems to be doing fine and even better than others. So yeah, maybe it's just a thing. I guess, yeah, you know, like this, that I wonder whether it's with having those different number of chromosomes, do you think that could also impact like how well it is able to infect other host species, or so many different hosts?
Well, I did some reading and research, and it depends on the organism. Sometimes having different numbers of chromosomes is seen as an adaptative advantage. So, like this specific strain may be resistant to a fungicide or something like that. But sometimes it's more like an accident, and it will be repaired in the future. So I'm not sure it will have an impact on pathogenicity. So that's why we are checking the content of these chromosomes and if they, the gene expression of them, and to see if we can find something that's common between the 2 of them that's not common for the rest of genome. So I'm not sure. I mean, that's a big open question.
That's good. Yes. So do you have any experience or knowledge of different Austropuccinia psidii biotypes colonizing the same plant or even the same leaf on a plant?
Naturally, well, I can say that like we have some field plants here in the city that, for example, sometimes a guava plant is on the side of a rose apple plant. One is infected and the other one is not. So for these two specific hosts, I can probably say that they are not infected, at least for this, like there is not cross infection for this host, these two biotypes, these two groups. I'm not sure about others because what we do is work with single pustule isolate. So we go to the field, collect isolates, and do a single pustule isolation in the lab. But this test that we are developing, we want to optimize it to work with mixed infections. So maybe in the future we can find more than one biotype in the same host species. But I never saw it, and it doesn't mean it doesn't exist. It's just that I was not able to detect it. Yeah.
Yeah, sounds good. So another sort of question, just when you were talking about the effectors and like the different the different or the different strains having different effectors in there, and then obviously and the effectors are being used a lot now, and just thinking about controls. So do you think that it's better to focus on the ones that are strain specific, or the ones that are shared with between all of the different strains?
I think it depends on the host. So for example, I think the strain specific, they may give the ability of specializing to different host species. So I think the strain specific ones would be better targets, yes, even for the biotype, because they are specific only for the pandemic and it's able to infect hundreds of different hosts. So I think it may have a role, but we know we have many other pathogenicity related genes that are not effectors that may have a role too. So I think I would start with strain specific ones, yes.
Yeah. No, I mean, and it's just, I think, like having that comparison, that ability now with all of the different genomes to look on such close detail to see like what these differences are. It's really, really, really tremendous.
Yeah.
So I think that that's probably it for today. And I have to say before I go, before we go, is that Thaís is joining us from Brazil, and it is very late at night, and her doggies have been very, very well behaved. So thank you again for joining us in your nighttime. So a video of today's webinar is going to be made available on the Beyond Myrtle Rust website in the next couple of days. And it's also going to be emailed to everyone who's registered. Our next webinar is going to be on Wednesday, May 22nd at 11 a.m. New Zealand time. And we'll hear from Michael Bartlett, who is a researcher at Scion and an integral member of our Beyond Myrtle Rust team, about the evidence for sexual reproduction in New Zealand populations of Austropuccinia psidii. So look out for the invite in your inbox next month. And thanks everyone. Hi, Rara. See you next time.